Hello, everyone. My name is Erin Newburn, and I'm the director of the Field Application Scientist team at Personalis. I'd like to thank the organizers for the opportunity to present today. I'll be presenting on our ImmunoID NeXT platform. It's an approach that's specifically designed for comprehensive tumor immunogenomics and advanced biomarker discovery. The landscape of predictive biomarkers continues to evolve, and it's becoming more clear that a single biomarker may not be sufficient identifying patient populations that are more likely to benefit from immunotherapies. Immunotherapy efficacy can be impacted by the tumor genomics, as well as germline genetic variation, and also the immune microenvironment. We see multiple studies that are beginning to expand beyond the current clinical biomarkers and into parameters such as gene expression signatures, germline and somatic HLA status, functionality of the antigen presentation machinery, tumor immune status, and others, with the goals of developing and identifying biomarkers with stronger predictive value. And as we move from conventional treatment modalities to these more personalized precision approaches in oncology, the information required to guide these treatments needs to be much more comprehensive in nature to capture the true biology of the tumor. We begin to see a shift from the smaller scale panels that mainly focus on cancer pathways and identifying mutations in the tumor that can be targeted to broader whole exome transcriptome assays looking at all 20,000 human genes to account for multiple biological systems. And this has been the focus at Personalis, that advances in our technology have enabled us to engineer comprehensive assays, whether tissue or plasma based, to better understand the tumor and immune interplay. And our ImmunoID NeXT product is developed to enable comprehensive characterization of the tumor, but also the immune-related components of the tumor microenvironment from a single tumor sample and a paired normal sample. We maintain a sample sparing approach and can work with just as few as 4 FFPE slides to derive both DNA and RNA, which we then can interrogate from multiple biomarkers. This scalable approach enables the identification of the biomarkers of today that you see listed here, but it also enables a path for novel IO markers as well. And by interrogating both the DNA and RNA features from the sample, we can begin to combine multiple biomarkers and produce more meaningful composite outputs with increased biomarker predictive power. And this is a unique capability of the platform. It's allowed us to identify composite neoantigen-based score NEOPS, which I'll discuss when I review our case study. And this is becoming the trend that we see in the translational space in an effort to better understand mechanisms of resistance and identifying patients who are more likely to respond to these different types of therapies and combination therapies. A more integrated multi-dimensional approach is needed. We know also that these are very critical, precious patient samples that have been acquired. Traditional processes have often meant and required multiple samples to be sent to perform multiple assays to get the data needed in the study. So for example, perhaps sending a blood sample for HLA testing, a tissue section for expression analysis, and then sending another tissue section for assessing mutational burden. The result is that sometimes investigators have difficult decisions, maybe a trade-off about which particular assay can be ran when there is limited sample, or dealing with complex data integration after working with several different platforms. At Personalis, we've created a more efficient process from sample receipt. We follow a dual extraction protocol, a unique library prep creation, and process the sample on our platform to produce a very data-rich set of analytics and multiple biomarkers, as you see listed here. So our ImmunoID NeXT platform was designed to be used as a singular solution, to use a single sample preparation, and to consolidate multiple biomarker assays into one. The ImmunoID NeXT platform is powered by a very innovative assay design. And first, it's by the depth of whole exome sequencing coverage, 300x mean coverage across the entire footprint, but also by our enhancement and augmentation. ImmunoID NeXT utilizes our proprietary ACE technology, accuracy content enhanced technology, that provides augmentation and coverage of these difficult to sequence genomic regions across the entire footprint. So more uniform coverage across all 20,000 genes as compared to a conventional approach. And I'll speak a bit more to this on the next slide. This is paired with our enhanced transcriptome approach of the tumor, where we deliver 200 million total reads. And this also utilizes our ACE technology, and we perform an exome capture enrichment approach for RNA sequencing. But secondly, within our innovative design, we provide these boosts of ultra deep coverage in critical areas of importance for oncology, with a boosted clinical grade sequencing coverage across a set of 247 cancer-related genes associated with solid tumors, as well as hematological malignancies, which enables also diagnostic capabilities from our platform. Other benefits within our design include the supplemental coverage of the TCR and BCR gene regions that allows investigation into the TCR dynamics. In addition, we have enhanced targeting of the HLA genes. We provide a variety of HLA-related analytics, including HLA typing, somatic variant calling, loss of heterozygosity reporting in the HLA loci. And we also provide MSI reporting as well as oncoviral detection. And this really makes this assay a first of its kind approach to really enable the accurate characterization of the predictive precision oncology biomarkers in both the tumor and the tumor microenvironment. And ACE, our proprietary chemistry, forms the technological foundation for many of the assays we perform at Personalis. And the problem with the standard exome and transcriptome solutions is that they often have gaps and inconsistencies in sequencing coverage. And this is something we have recognized and addressed with our chemistry. This is a visualization here of those gaps that you see across a particular gene. I like to refer to this as the peaks and valleys, areas very sufficiently covered, maybe others that are suboptimal. And then this is a nice visual of the quality of the data you would receive from an augmented exome approach such as ACE. We provide with the blue, basically what you would get from a standard coverage approach, plus the green, the supplemental coverage demonstrated here in this sequencing coverage plot. Our goal is to really achieve more accurate, more uniform coverage gene-wide. So the gaps where you see no blue coverage and the sequencing depth are now filled in by this enhancement. And we do the supplementation for all 20,000 genes with ImmunoID NeXT. In our Personalis clinical lab, we receive a variety of different samples, FFPE, whole blood, fresh frozen, PBMCs. Shown here is a snapshot of some FFPE materials. And you can see that this is the reality of working with clinical FFPE materials. They come in a variety of different qualities and quantities. In our lab, we have specifically optimized to work with these more challenging sample types. We currently have a greater than 90% success rate for any sample we receive in our lab. And this data shows the percentage of samples passing our DNA and RNA extraction QC criteria here in the right. These samples come from many different biomarker and personalized cancer therapy customers and many different sample collection sites. And as I mentioned, we can work with just as few as 4 FFPE slides. A common challenge in oncology biomarker discovery is taking on the feat of analyzing the data generated from an exome and transcriptome solutions. And we've created various analytical modules to allow our customers to generate informative immuno-oncology insights from the very comprehensive DNA and RNA data that we deliver with ImmunoID NeXT. And these algorithms enable us to look at the broader cancer ecosystem. We have co-developed these solutions to make sense of the data with the assay development, and we report on multiple biomarkers. We have developed DNA, RNA pipelines that we mine for various outputs. We've optimized for HLA. We developed a neoantigen prediction pipeline trained on immunopeptidomics data, as well as other analytical capabilities to generate a very comprehensive data set to support the translational studies of biomarkers. In the next section, I'd like to illustrate how our ImmunoID NeXT assay and informatics can really be put to use by presenting you with a recent case study performed by our R&D team. And it shows how ImmunoID NeXT can be utilized to understand mechanisms of resistance, how we're able to derive some insight on responders and non-responders using our high content approach. And so this study is conducted in collaboration with Dr. Sekwon Jang from the Inova Shar Cancer Institute. And to describe this cohort, we obtained samples from late-stage unresectable melanoma patients who had received anti-PD-1 therapy. These Patients all received anti-PD-1 therapy at the Innovashar Cancer Institute in Virginia. They were then evaluated according to recess criteria. We received the pre-treatment FFPE tumor blocks along with the corresponding blood sample. We ran our broad immunogenomic profiling assay of ImmunoID NeXT, and then we're able to take the molecular data in conjunction with the clinical response data to look at and profile potential mechanisms of response and resistance. So we first looked at the mutational landscape of the cohort, and as TMB is a key biomarker in melanoma, we started by looking at the tumor mutational burden. And with our platform, TMB was optimally calculated from the whole exome footprint, as opposed to using a smaller scale gene panel, where the TMB must be bioinformatically extrapolated. And we reported on the SNVs and indels using the exome footprint. We observed a slightly higher TMB in responders than in the non-responders, but there was overlap between the groups, suggesting that in this particular cohort, TMB was not a strong predictor of response and did not show strong association with progression-free survival. And since TMB provides a look at tumor foreignness, it's basically a surrogate biomarker for immunogenicity, we also wanted to look a bit further at mutational landscape and compute the neoantigen burden as a next step. This was done using our internally developed pipeline, our neoantigen ID. And when we calculated neoantigen burden in this cohort, we found that it was more strongly associated with response and improved progression-free survival with more significant p-values. But we still observed some unique profiles that we were interested in exploring further through our immunogenomic characterization with our platform. And I'll highlight a few cases next. So this first example was a partial responder, but with a higher neoantigen burden. And we looked at multiple parameters to explain this profile and found alterations in the HLA data that might play a critical role. So from the patient's HLA typing, we saw this individual was heterozygous for all three of the HLA class 1 genes. And this partial responder had two distinct alleles from the HLA typing results. But when we looked at the tumor, we found that there was a chromosomal deletion resulting in the removal of one allele in all three of the A, B, and C genes. And so this HLA loss of heterozygosity event is potentially detrimental to the number of neoantigens that could be presented for immune surveillance and may explain the partial response to immune checkpoint blockade. We're also curious about another patient in the non-responder group who had, again, a relatively high neoantigen burden. In this patient, we detected somatic mutations impacting two HLA class 1 genes, HLA-A 0201 and HLA-B 1501. Both mutations had a high allelic fraction, high variant effect impact, suggesting that they might be critical in affecting the function of the HLA molecules. And in addition, we predicted which HLA the patient's neoantigens were likely to bind to. And we found, in fact, that 40% of those predicted neoantigens were predicted to bind to the two mutated HLA alleles. Again, although these neoantigens were present, found at a high allelic frequency, they failed to be effectively prevented by the HLA. And this could be a potential mechanism of immune evasion and possible explanation of the lack of response in this case. And in another patient, a non-responder, we also observed a somatic frame shift variant in the beta-2 microglobulin gene. Of course, B2M is a key component of the antigen presentation complex, and this frame shift variant potentially limits correct HLA class 1 folding and antigen presentation for the class 1 peptide MHC complex. And this is consistent with other studies in the literature showing a similar mechanism of resistance. So as you can see so far with our approach, as shown here in the case study, we can report on TMB, neoantigen burden, but we can also integrate these molecular features to create a more composite biomarker, a composite score. And as we see from the literature, many groups have shown that improved biomarkers predicting immunotherapy outcomes will likely need to be much more integrated and multidimensional to really capture the crosstalk between the tumor and the immune system. And so when you're counting non-synonymous somatic mutations in TMB or neoantigens in the neoantigen burden, it's really a first step. if these neoantigens are not able to be properly presented to the immune system for surveillance, how relevant are they in the assessment of the mutational landscape? And we saw that when we look and took into account the HLA mutational status of the patient, so here we looked at HLA loss of heterozygosity, somatic mutations in the HLA gene, and also B2M loss, and we removed those neoantigens predicted to bind to those mutated alleles and adjusted the antigen burden, we found this yielded further significance when looking at response and non-response. So this adjusted neoantigen burden, which we refer to as NEOPS, was able to better parse out responders and non-responders in our melanoma cohort. And this is just one example of what we enable with the platform and the various analysis, how we can integrate features to give some insight that can help identify novel and more meaningful biomarkers for different cancer types. So this case study again demonstrates how just a few of the analyses that are possible and enabled by our platform, the various analysis and insight that can help produce more relevant biomarkers for different cancer types. Another assay I would like to highlight today is the NeXT liquid biopsy exome scale assay. And our strategy goes beyond where targeted panels are today to provide a much more comprehensive view of the tumor heterogeneity using liquid biopsy approach. So NeXT liquid biopsy, it's a less invasive method to identify novel biomarkers. We have taken a different approach than the standard traditional panel-based liquid biopsy assays and have developed an exome scale approach, sequencing at high depth with an average coverage of 2000x across the entire footprint, but also including a significant sequencing boost for 247 cancer-related genes, and those particular genes are sequenced at 5000x. Also, in addition, we have innovated at the computational level to eliminate bias and reduce errors due to background noise from the plasma. And so keeping with the comprehensive themes of our assays, in the first section, we discussed how we provide the full picture of the solid tumor through immunogenomic profiling with ImmunoID NeXT. But now, in addition, we also have an exome-wide look of the plasma as well. And so some of the key applications we look to enable from the plasma are in understanding the biology at baseline, but also in the context of the evolving tumor dynamics in response to therapy, such as understanding how and why acquired mechanisms of resistance emerge, clonal and subclonal evolution, how biomarkers and mutational signatures can change, and how neoantigens can change over time. And we believe the plasma assessment is complementary to the tissue biopsy in capturing the true biology and heterogeneity of a tumor. And so when you're trying to build this systemic view of the disease, it's ideal to have both the liquid and the tissue biopsy because there are elements of the biology that you can only see with one or the other. So when looking for this full characterization of the tumor, the tissue does remain important because you can access important information about the tumor microenvironment. You can robustly interrogate the RNA for gene and immune expression signatures. You can have very sensitive fusion and copy number variant detection. You can also look at the TCR alpha and beta clonality coming from the tissue and other important biomarkers. But in conjunction with the tumor, the liquid biopsy is also important because it is more representative of the inter and intratumor heterogeneity. And the assay that we have developed is configured to profile the tumor and the matched normal and the plasma as a set to identify somatic variants and de novo events from 50 nanograms of cell-free DNA. So we're using a tumor-informed approach with our current configuration, requiring the tumor, the normal, and the plasma sample from the patient. And we have demonstrated exceptional sensitivity of the assay. We achieve over 95% sensitivity for SNVs and indels in the boosted region for variants at a 0.5% minor allele frequency, and similar numbers when we look exome-wide for variants at a 1% minor allele frequency and above. And so for our next liquid biopsy approach, there are several different study configurations that can be supported by the integration of NeXT Liquid into your assays. Having the information included at baseline is very powerful, but also allowing the liquid to provide information at various time points, looking at monitoring changes at various time points from that patient sample. So this integration allows for a very comprehensive characterization by complementing the information gained from the solid tumor specimen. So in my concluding slide, with the platform we have established, we really are a partner throughout the development process, throughout drug discovery. And currently we are supporting many pharma and biopharma customers in their clinical development and translational research of their phase 1 through 3 clinical trials. And this started with ImmunoID NeXT. We've now enhanced this with our NeXT liquid biopsy offering. We also provide clinical testing support with our NeXT DX LDT. We did not focus on this assay today in the presentation, but the 247 genes that I mentioned that we enhance and boost to very deep clinical grade coverage, as we can provide a clinical report to the ordering physician from those genes. And then based on that tumor profile, the report delivers the relevant therapy recommendations and clinical trial matches. And this is another very exciting element for us at Personalis, as it represents our first step towards a companion diagnostic. And this is precisely what we are doing at Personalis with the development of our NeXT CDX. I also want to quickly mention here on the front, we do also provide various research solutions, such as configurations for RNA sequencing, microRNA sequencing, to support the early discovery and preclinical work of our pharma partners as well. So thank you very much for your attention today. I hope that gave you some insight into our platform and how we are taking on the challenges in biomarker discovery at Personalis. Thank you.